So this is the lesson 13 group problems for genetics. So for the first problem, we are supposed to show like what the banding pattern would look like on the gel. So I drew a picture right here. For the first part I did was rewrite the actual sequence, and then one at a time. Since the first one's a C, C would be the furthest down because it would be the smallest, and then you just keep working your way up until you get to A, which would be the one closest to the top because it would be the biggest. And then you just put them in the right categories. For part B, it says, what if we use DNA polymerase 1 from E. coli instead of TAC? It probably wouldn't work because the high temperature would denature it. For part C, if you accidentally added E. coli to helicase, I said it would work normally just because it already unwinds by itself. You don't need helicase, so it should be fine. D says you add DT-ATP in one of the 4 twos, but you forget the others. So you would only show 3 bands. The full band, and then with the other 2 A's, because it would stop at each of them. So you wouldn't see any of the other banding patterns on G, T, or C. Part 2 is just fill in the blank. During a typical PCR, the third step is done at 72 degrees Celsius, while the second step is done at 55 degrees Celsius. Ampicillin resistance is a common selectable marker that's found in many plasmid vectors, while restriction enzymes recognize and cleave DNA molecules at palindromic sites. Part C, during agarose gel electrophoresis, DNA is placed in an electric field. DNA migrates towards the positive end, and DNA migrates towards the positive end during electrophoresis because DNA is negatively charged, so it goes towards positive. So in my number 1, I should have switched the positive and negative for my picture. For 2 part D, DD-ATP, that's the 2 OH group and the 3 OH group, so it's missing both hydroxyl groups. Part E, transformation is a term used to describe the process when bacteria uptake DNA in the environment. And then part F, during blue-white screening, IPTG is commonly used to induce expression of the lac operon, while XGAL is cleaved by beta-galactosidase to turn E. coli cells blue. Part 3, I didn't really understand. I asked for help a while ago, but I never got it. But I'm guessing that we had to look for a palindrome and only found one. It was the CAA-TTG in the 5 to 3 direction, and then also it's complementary. So I wasn't really sure what to do with this one. For part B, I just counted all the T's and A's and or C's and G's and took the proportion out of the whole, and then I just plugged it in. There's 24 nucleotides in that gene. So I got 72.2 degrees Celsius. For part C, if we include DD-CTP, then the reaction would terminate early and we wouldn't get the full strands because it would stop as soon as we hit a C. And then part D, we learned that DNA replication E. coli. What about DNA A and DAN? Why are these 2 proteins not included? I said because of the high temperature, they'd most likely denature. Part 4 gave us 6 different reactions and the fragments it produced based on the restriction enzyme that was used. So part A, I just drew the map. So the first column, or column 0, is all the standard lengths, and then 1, 2, 3, 4, 5, 6 just corresponds. Part B, we had to map it. So I got from the starting point, you go 50 units, and you get your BAMH1, another 150 units, you get your EcoR1, go 100 units, and you get your HEN3, another 100 units for your BAMHI, and then another 500 units to hit your N. And all the fragments you get, if you put all 3, would be 50, 100, 150, and 500, and you'd get double the amount of 100s because there's 2 100 fragments. For part 5, it says, how would we generate cDNA from mature mRNA to clone a cDNA version of YGF into a plasmid vector and use blue-white screening to identify plasmids that contain the YGF cDNA? So I'd say we'd mix the mRNA with the primer, a poly-DT primer, so it could recognize the poly-A tail on the mature mRNA. Then you add some reverse transcriptase and DNTP, so it could start making the DNA, and now you have an RNA-DNA hybrid. Then you put RNA ACE H to eat the RNA, and then just DNA polymerase 1 to synthesize the second strand of DNA. So now it's double-stranded DNA. And then you put your DNA with a restriction enzyme that recognizes the palindrome with the YGF gene and a vector. And you also need DNA ligase to seal it up so it gets into the plasmid. And then you have to use transformation to get it inside the bacteria. And some of them will have your gene, and now some of them won't. To identify it, you're going to add IPDG to start the LAC-Z gene. And then you're also going to put in XGAL. And all the ones that turn blue, don't have your gene, and all the ones that are white would have the gene. And now you can, separate them or grow them on a different plate, so you can just have the ones with your gene. And then the last question asked to describe CRISPR-Cas9. It's a gene editing tool, so it's able to take the RNA and give it an RNA sequence, and it will find the DNA that matches it, and we'll be able to cut the DNA on both strands. And then when DNA tries to fix itself, it messes up and it could cause mutation. Or sometimes you can put another repair template in, so it will add a gene. So you can cut a gene and mess it up, and then add a gene in there so it can have a new gene. And this technology came from bacteria. It's how they fight viruses. When they get a virus, they keep a copy of that virus. So when it ever comes again, they can fight against it by cutting it and messing it up. And scientists plan to use this to help with medicine or agriculture and trying to fix human diseases. And Cas9 is an enzyme. That's what it is. And that's it for the lesson 13 group problems.